Hello students, now we will start with new topic that is about hexoses. In our syllabus we have two examples. One is glucose and another is fructose. Glucose formula is C6H12O6 and fructose formula also same, that is C6H12O6. But there are some structural differences between these two compounds. Hexoses, they exist in two forms, that is open chain form and also ring form. Open chain form also called as linear form. But the names for the ring forms of glucose and fructose, they differ. First one, if you take glucose, open chain form or linear form is like carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So here there is aldehyde group at carbon 1 of glucose, and at second carbon there is OH, H, H and OH, OH and H, OH and H, OH and H and H. Now, in case of living organisms, the ring forms exist to involve in the metabolic reactions, not the linear or chain forms. These linear or chain forms we are drawing for our convenience, but naturally the linear or open chain forms will not be existed. Always these hexoses are involved in any type of metabolic reaction only in ring forms. So glucose, now if it has to get converted into ring form, how it is changing or its structure, or what are the modifications that are done within the molecule. So there will be a bond between carbon 1, which is having this aldehyde double bond O group, and carbon 5. It will remove one water molecule which again will be dissociated and gets connected in the ring form. That sequence I will draw here now. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. The remaining distribution of OH and H groups are same. But between carbon 1 and carbon 5, there is formation of one bond, and the hydrogen that has to be present, it will be binded to the OH group at 5th position, and initially it is removed in the form of one water molecule. But immediately this one gets dissociated into H plus and OH minus ions, and again they will be reunited in the ring form. So coming to the ring form, the structure of the ring form will be like this in case of glucose. As we have said that there is one bond with that of oxygen between carbon 1 and carbon 5. So I am writing oxygen here and carbon 1 here, carbon 5 here. So according to the formation of bond, the ring form is correct. So, this is carbon 2, carbon 3, carbon 4. Then what about carbon 6? This will be fitted at carbon 5 in the form of a branch. So this will be the sixth carbon position as a branch to the ring form. Now, this dissociated H plus and OH minus ions, again they will be attached at carbon 1 position like this, and at carbon 2 position there is OH and H. At carbon 3 position OH is on other side, so I am writing OH here and hydrogen here. At carbon 4 again there will be OH and hydrogen here. At 6th position there are 2 hydrogens, 1 OH group, that I am writing in the form of CH2OH. 1 carbon, 2 hydrogens and 1 OH group. The ring form of glucose is called as pyranose ring. Since the bond is formed within the molecule and between carbon 1 and carbon 5, since such type of compounds which have formed a bond between them or linkage between them are called as hemi compounds, but here this is an aldehyde group. So we are giving for the glucose molecule as hemiacetal compound. Hemiacetal compound, since carbon 1 position of glucose is having aldehyde group. But in case of fructose it is not so, because fructose at its second carbon it is having ketone group. So there we cannot use the term like hemiacetal. There we can use a term after formation of the ring form from the fructose as hemiketal compound. Now, there is another term for hexoses commonly, that is polyhydroxy compounds. This polyhydroxy compound is applicable for both glucose and fructose because the compounds are having more number of OH groups. Even fructose also will be having more number of OH groups. When I draw the structure, I will show you. So, because of existence of more OH groups within the compound, hexoses are commonly called as polyhydroxy compounds. So, here we have seen formation of pyranose ring. Why we are calling it as hemiacetal? Because between the carbon 1 and carbon 5 of the glucose molecule, there is formation of linkage or the bond with the help of oxygen here. So we are calling it as hemiacetal compound. The formula is the same. So we will represent the open chain form of fructose: carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon 6. Here at carbon 1 there is normally OH and H groups like this. At carbon position 2 there is one ketone group. At carbon position 3 again same like that of glucose. Like this. Now again here in case of fructose also there is a bond formation between carbon 2 and carbon 5, but now temporarily this OH group is removed from here and it is attached at carbon 2 position, because at here now the structure is going to change its angle, means it is going to bend like this. That is how. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. When there is formation of a bond here, the distribution of all the other compounds is same. Groups are same. So it will be considered as CH2OH. But here, this should have one more bond here, because 1, 2, 3 are only existed at the second position. OH, NH, NH. Now, since the second carbon is lacking one more hydrogen, the OH group that is removed here from fifth carbon will come and again bind here at carbon 2 position, so that the number of carbons, hydrogen bonds are same. 1, 2, 3, 4. For this carbon also 1, 2, 3, 4. It is quite common here in case of fructose. Now, actually, in case of living organisms body, as I have told you in case of glucose, the open chain form or ring form will not be existed. 99.9%, we can say 100% of the carbohydrates are existed in ring forms, either in pyranose ring or furanose ring. So, I told you in case of glucose, the ring form is called as pyranose ring. Then how this compound is existed in the ring form? Actually, here it is not in this form. We should not write like this. We should directly express in the form of ring. But just to tell you how the ring has obtained, I have drawn this structure. If you consider this structure like this, there is a oxygen or bond between carbon 2 and carbon 5. So I am representing carbon 2, carbon 5 here and one oxygen here. Carbon 2 is having one branch-like structure of carbon 1, where the compound is CH2OH. Now, what about this OH? This OH we can represent here. Now, at fifth carbon also there is a branch with help of carbon 6. Again here also CH2OH is existed, CH2OH, CH2OH. That I have written here. Now, what about carbon 3 and carbon 4? Carbon 3 is having hydrogen at one side and OH on other side. That I am representing as hydrogen here, OH on other side. In case of carbon 4, the arrangement of OH and hydrogen will be like this. So, the ring form has been obtained from the fructose open chain form. Name of this ring form of fructose is called as furanose. Furanose ring. Since the bond is formed within the molecule, but this compound is a ketone compound or ketose sugar, we have to give a term like this furanose ring form or furanose is a hemiketal compound. In case of glucose, I told you there is one aldehyde group at carbon 1 position. So we are giving it another term for glucose as hemiacetal compound, since glucose has formed a bond within itself. But there was a fact that glycosidic linkages are not formed or existed either in glucose or in fructose. So here I have told you few different terms for hexoses. Hexoses are also called as polyhydroxy compounds because when we take few examples, even if you take galactose also, there are many number of OH groups. So, monosaccharides like glucose, fructose, galactose, all these are 6 carbon compounds and they are having many OH groups, so called as polyhydroxy compounds. Example, glucose, fructose, galactose. In case of glucose, carbon 1 position is occupied by aldehyde group. But the linkage during formation of pyranose ring is between carbon 1 and carbon 5. There, since the aldehyde group is existed, we are giving a term like hemiacetal compound. But in case of fructose, the bond is formed between carbon 2 and carbon 5. But the thing is between or within the molecule. So here, since it is a ketone, group is existed at second carbon, we are giving another term for fructose ring form as it is a hemiketal compound. Let's compare both hemiacetal compound or the pyranose ring form and hemiketal compound of this furanose form or hemiketal compound. Here it is hemiacetal compound. Now, one more point. The pyranose ring or hemiacetal form of pyranose ring, it is having how many sides? 1, 2, 3, 4, 5, 6. So it is also called as hexagonal ring. Hexagonal ring. The name is given because of existence of 6 sides in the ring. 6 sides. In case of furanose ring, how many sides are there? 1, 2, 3, 4, 5. So it is also termed as pentagonal ring. And some way this pentagonal ring, it resembles the pentose sugar ring forms like ribose and deoxyribose, etc. One more difference if we observe, the ring form is having only one branch. Pyranose ring form is having only one branch, that is at carbon 6 position. But furanose ring is having 2 branches. That is at carbon 2 position and carbon 5 position. What are the branches? Carbon 1 branch and carbon 6 branch. So these are few points regarding pyranose ring of glucose and furanose ring of fructose. Next we will see about pentose sugars. So next topic is pentoses. So what examples can we consider here? One is ribose sugar. Next is deoxyribose sugar. Next is arabinose. Ribose is pentose sugar in RNA. Deoxyribose is pentose sugar in DNA. Arabinose is abundant pentose sugar in nature. It is existed in the outermost layers of vegetables and rice husk, etc. In case of bacteria, the main source of energy is obtained by the arabinose type of pentose sugar. So, it can be considered as main source of sugars for bacteria. Now, coming to ribose, how the structure is going to be? In case of ribose, the structure is going to be like this. Carbon 1, carbon 2, carbon 3, carbon 4. How many sides are there? 1, 2, 3, 4, 5. Now, but in the ring, there are only 4 number of carbons. The 5th carbon is considered as the branch. Now, if we distribute the hydroxyl and hydrogens here, the arrangement will be like this. Now, in case of ribose sugar, this oxygen at second position of the carbon is considered to be unstable atom because it is having a different nature of binding with any type of metals or easily undergoing hydrolysis in presence of water. Only the oxygen at second carbon of ribose sugar is having such a nature. So, total RNA molecule is behaving as an unstable compound, or pentose sugar, that is ribose, is considered as unstable sugar. Unstable sugar. That term is also given because of oxygen at second carbon position. Now, this oxygen easily binds with any type of metals and undergoes oxidation. Next, this oxygen is easily prone to hydrolysis. So, it gives a nature to the total RNA molecule to be in an unstable nature. So, during the course of evolution, we know that the first genetic material is RNA. If RNA we are considering as a genetic material, and if it is easily changing its shape, then what happens in course of evolution? The changes or the modifications or the variations in living organisms will be unlimited, uncountable. See, to predict also it will be very much impossible. Organisms, they try to adapt to the new environment. They take some time, but within that time period, again if the RNA is undergoing any variations or unstable natures, or if RNA is responsible for changing of the shapes of the total organism's structure in morphology, in anatomy, etc., then it is very impossible for the organisms to survive in nature. So, to prevent that, what RNA as a genetic material has done, it has removed this oxygen from the pentose sugar and made the compound as stable compound. Now, how this and where, in which type of organisms did this RNA has removed that oxygen means, we can take few examples like, one is inside nucleus. Inside nucleus, RNA has changed the structure of the pentose sugar by removing the oxygen. One example. Another one, inside chloroplast. Inside chloroplast. Another one, inside mitochondria. Inside mitochondria. Another one, in DNA containing prokaryotes. In DNA prokaryotes. Now, how? Already DNA is there. No. It is like previously there was genetic material RNA in some types of prokaryotes. Later that was modified into DNA, that is stable compound. Now, here I have given here one point, easily binding to any of the metals. So what is the major metal that is present inside the nucleus? We can take one best example as zinc, which is providing the coenzymes for the functional activity of the enzymes during the cell division. In chloroplast, what are the metals present? Magnesium is present within the chlorophyll molecule. Manganese is present in the inner thylakoid membrane during water molecule breakdown. And in mitochondria, what are the metals present? Ferrous is present. So, and in case of prokaryotes, we know that any type of ferrous, manganese, all will be present. Zinc also will be existed, which is supporting in cell division. So, basing on such types of important concepts, we can tell oxygen at second carbon is creating instability to the compound, so that the total instability is applied to the RNA, which is considered as a genetic material. Now, here I have written one point that it is easily undergoing or easily prone to hydrolysis. Hydrolysis in the sense addition of water molecule and breaking the compound. Hydrolysis means we are not adding only one single water molecule, we are adding a group of water molecules. We know that water molecule will be again dissociated into H plus and OH minus ions. These forms are existed in the water molecules. Now, when few number of H plus and OH minus ions, when few number of these compounds are taking out the oxygen out along with the hydrogen, one molecule of H plus ion will be replaced here. Then we can call this compound as deoxypentose sugar because here oxygen was removed. Okay, so this is about the ribose sugar. Now, what is the structure of deoxyribose sugar? The structure of deoxyribose pentose sugar or deoxyribose sugar is like this. So, carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. Carbon 5 is considered as the branch. Now, again the same structure, but here instead of OH, as we have seen how the oxygen or OH group is removed with the help of water molecules, it is existed only with hydrogen at a second carbon and again hydrogen here, but at third carbon there is OH, H, OH and here CH2 OH is present at fifth carbon position. Now this is nothing but under pentose sugars. Now when oxygen is removed from the ribose sugar, so we are giving a term called as deoxy. Since oxygen is removed here, so called as deoxy, but the remaining structure is same like that of ribose. So deoxyribose sugar of DNA. Now this is considered as stable sugar or stable compound, which provides complete stability for the total DNA. that provides stability for DNA. Means if any another metal has come nearby also, the structure is not going to break down its bonds which are present within them for total DNA. Means another, if any water molecule has come nearby, still the bonds are not getting broken down. The structure is being strictly maintained by the deoxyribose pentose sugar.